All right, so when we talked about bones, we talked a lot about how it's a scaffold for attachment for muscles. And muscles, of course, are the major component for how we move. But not all muscles are associated with the skeleton. We have other muscles which are important for our vital functions, including what makes up our heart. Our cardiac muscle is what we find in our heart and has specific characteristics as we've gone over before. And our smooth muscle is important for all other functions, such as moving things through our digestive system and regulating blood flow through our blood vessels. But when we think of muscles, we often refer to those skeletal muscles, which are abundant in our body. And I had to include this picture, although this looks somewhat real. This has to be fake. I do not believe anyone can actually get to this size. So again, going back to our muscles, it is one of our four major categories of tissue. And its unique properties include that it is excitable. That means it can be triggered in response to signals sent by the nervous system. Mostly the nervous system. There are some actually excitable muscles which can act without the nervous system input. They are also able to contract and shorten in length, and the actual excitability causes them to contract. There is actually no action for lengthening, but relaxing the muscle will cause it to lengthen. They can also stretch, so there is this elastic property to them, or stretchability to them. And then after they are stretched, they can return to shape, and that would be the elasticity of them. The three main functions of our muscles include motion and movement, not only of our body, but of course of other things, to maintain our posture and to produce heat. So when you are cold, your muscles may shiver to produce heat. We have already gone over the three types of muscle tissues. Our skeletal muscle is striated and is under voluntary control, conscious control from the nervous system and brain. The cardiac muscle is striated but is under involuntary control, and so is the smooth muscle, but smooth muscle is also unstriated. So we'll first talk about skeletal muscle. Skeletal muscles as an organ, as a whole, can attach to bones or have to attach to bones at some point. They can either do this directly, where the epimysium, which we haven't talked about yet, is fused to the periosteum, which we have talked about, the outer layer of bone. And it can also attach to parts of the skeleton indirectly through a aponeurosis, where the muscle attaches to another muscle which attaches to the bone, or through tendons, where dense connective tissue attaches to the muscle and the bone. There are more than just muscles in muscles, right? So muscle is the largest component, of course, found in muscle tissue anyway, but there is also arteries, veins, and capillaries, which are providing blood supply to the muscles. There are nerves, which are important for causing that excitation to occur and sending feedback from those muscles. There is, of course, muscle tissue, and then there's connective tissue holding it all together. Skeletal muscle also has sheets of fascia, which are dense connective tissue which encapsulates all the skeletal muscle in 3 different layers. The 3 layers are the epimysium, which is the outer part which surrounds the whole muscle itself. So you may have a muscle such as the gastrocnemius. It has that fascia layer, epimysium, around it. And then within there is the perimysium, okay, which surrounds each fascicle. And the fascicles are also surrounded by, or can be subdivided into fibers. The fibers are covered by endomysium. And then the endomysium has many different muscle cells within it. The function of these mysiums is to keep the muscle from overstretching and allow it to return to shape. It encapsulates the whole thing, and it also can provide a passageway for blood vessels and nerves to individual cells or individual fibers. The properties of fascia, or properties of meat which affect fascia, include the stiffness and rigidity of the muscle. So meat that is tender, that is cooked just right, has this fascia layer broken down. Okay, so the amount of dense connective tissue and collagen within the meat is going to, and how it's broken down, is going to affect the tenderness. Younger animals have less of this connective tissue because their muscles are still forming and taking shape. And so pigs, which are slaughtered at a younger age than cows, generally have more tender meat than the cow meat. So pork ribs are generally more tender than beef ribs. You can tenderize your meat mechanically by pounding it with, here's a meat tenderizer here. Kind of breaks up those fascia layers. Or you can use this meat tenderizer enzyme, which breaks down that fascia. Or you can just cook it longer at a lower temperature. All right. We're going to then get into a microscopic layer and look at the muscle cells, which are also called muscle fibers. Okay, so this is a muscle cell here. Sorry, this is a muscle cell, this whole thing, muscle fiber, which is made of many myofibrils, which is the organelle. The muscles fibers themselves are larger than most cells. They can be 10 to 100 micrometers and up to 30 centimeters long, which is about a foot in length. And they have multiple peripheral nuclei, and then the myofibrils are then kind of squished all together within the endomysium. The myofibrils can be broken down into different parts. The myofilaments, these are the proteins. Now we're looking at the molecular level. You have thick myofilament and thin myofilament, which are actin and myosin. Okay, and so you can see on this figure, the purple here is the thick filament, which is myosin, and then this green part here is the thin part, the actin. All right, looking closer at the muscle fiber, there are lots of structures around there. So you don't just see myofibrils, you also see some other things, including the sarcolemma, which is the plasma membrane of the whole muscle fiber. Okay, there it is, sarcolemma. The parts of it will actually extend into the myofibrils, and those are called T-tubules, and these will be important. We'll talk about them later. The sarcoplasm is the cytoplasm of the muscle fiber. Okay, so that's surrounding all these myofibrils. The myofilaments, we just talked about, the thick and thin, actin and myosin. The sarcomere is the functional unit of the muscle. So this is, one sarcomere is where the actin forms this connection called the Z-disc to the next Z-disc, and there is a length of myosin within there. And then we have the ever important sarcoplasmic reticulum, which is smooth endoplasmic reticulum, which acts as calcium storage, which is important for the excitability of the cell, of the organelle. When you break these into different parts, they're all labeled here: Z disc, H zone, A band, I band. What do these mean? Well, the Z line is the anchor for the thin filaments where they are connected together. The M line, or the middle line, is just the anchor for the thick filaments, and it does not actually have it's not actually a physical structure, but just kind of an imaginary line. The A band is the length of the myosin, and the I band is the length across the sarcomere, which only has actin. And then the or the H zone, or the Healy zone, is the area of myosin where there's no actin overlapping it. Okay, and these, the M line is in the center of the H zone, and these, you can see these on the sarcomere, and they change in length depending on whether the muscle is contracting or relaxing. Okay, so here's the figure from the book. The M line is right in the middle of the A band, where you have only myosin. The lighter band is where you have only actin. The Z line is that anchoring point. The H zone here, or Healy zone, is where you have only myosin. Sorry, the A band is the complete length of the myosin. And then the I band is from one end of the myosin to the next, or the area where you only have actin. Okay, and then from Z line to Z line, that is the sarcomere. Now, on each of these, you also have actin, you have troponin and tropomyosin, which we'll talk about in a second, and then these myosin heads. When the sarcomere contracts, okay, so what happens is the Z lines are going to come closer together. So what happens then? Well, you also see the I bands shorten. The distance between the Z lines shortens. The H zone, it shrinks to almost nothing. And the A bands move closer together, but they remain the same length. Right? So this A band from one to the other is going to be closer together. All right. And this is an unlabeled thing we'll look at, or something similar in lecture. But a more simple diagram of these, what happens in a relaxed and then contracted sarcomere. Now, we mentioned the sarcoplasmic reticulum, and it's ever important. It is an important part of the system that allows for excitability. It is, again, a smooth endoplasmic reticulum that goes all the way around the circular myofibril. Most of them run horizontally, but there is, and that allows the communication along the H zone. But there are sections which are thickened, called the terminal cisterns, and they run perpendicular, so they are running up and down. See, there's a terminal cistern here, and here are the horizontal sections. Closely associated with the sarcoplasmic reticulum are mitochondria and glycogen granules, and those allow for energy to be available for the actin and myosin to contract. And again, its main function is to store and release calcium to initiate that contraction. The T-tubules then are parts of the sarcolemma, which extend at the A band, I band junction, and is sandwiched between two of the terminal cisterns. And so those three, the ends of the two terminal cisterns and the T-tubule, are called a triad, and they go all the way around the sarcomere, and that allows the signal from the sarcolemma to be transmitted to the sarcoplasmic reticulum so that it can release calcium. All right, the structures on the actin include troponin and tropomyosin. The troponin acts as basically an anchoring point for the tropomyosin, and the tropomyosin covers binding sites, okay? And those binding sites are covered only when it's inactive. To activate the contraction between actin and myosin, those will shift and expose those binding sites. Myosin then have these heads, okay, where they have actin binding sites, so that's where it connects to the actin, and ATP binding sites, where they can attach ATP, which can be used for energy. And they also have the ability to break down ATP, ATPase, which would be the enzyme important for breaking that down to use it for energy. A cross bridge is formed when the actin binding site attaches to the myosin binding site in the actin. All right, so now we're going to study, well, we know all the parts. How does this work? How does a signal lead to excitation, lead to contraction? Okay, the first part is you need, for skeletal muscle, to have a signal. You need the nervous system to say, all right, start contracting. And so you have on one end the end of a neuron attaching to these muscle fibers. And it's an electrical signal, so it's we all know our brains operate on electricity. The signals they send are changes in voltage, and that's going to then initiate the contraction mechanism. But the actual contraction itself is a mechanical action. It's a physical force where something's actually moving. So you're converting electrical signals to mechanical action. So induction is the first step of contraction, and this involves the stimulation of that muscle fiber. Okay, and then that change in membrane potential allows that excitation to go down into the muscle cell. An electrical current, which is called an action potential, we'll study that more in the nervous system, first has to be not created but initiated into the sarcolemma. And then it is propagated along the sarcolemma and then down into the T-tubule, and that's where it triggers the sarcoplasmic reticulum to release the calcium, and that's the signal then to for the actin and myosin and the troponin and tropomyosin to shift. So the neuromuscular junction is the point at which this happens. So this is pretty similar to what the figure I just showed you, except this is one that I drew, and we'll go over these different steps. But first we're going to review, so here's the neuromuscular junction, the neuron and the muscle where they meet. But first we're going to review the electrochemical equilibrium. As you remember, most cells have a negative resting potential of -70. Muscle cells have -85, and that is fueled by these large negatively charged proteins which cannot leave the cell, and balanced by the attraction of positive ions to these negative proteins and their ability to move through the cell membrane based on leaky channels and sodium-potassium pumps. All cells have these passive channels or leaky channels, but muscle and nerve cells also have these active channels, and that's what makes them excitable. An action potential then is a temporary change in that positive and negative that charge and then ending with the resting potential. Active channels are usually closed. There's two types: voltage gated, okay, which takes a charge in order to propagate it, and chemically gated, which requires some sort of chemical to allow them to transfer. And the chemical that opens the gate is called a neurotransmitter. Okay, so a neurotransmitter would then bind to this chemically gated channel that would then open and allow substances in. A voltage gated one would then require a electrical signal to open, and then that would allow substances in. So voltage gated channels open and close when there is a change in the charge of the cell. They are closed at the resting state or resting potential, and they open when this -70, well, it'd be -85 in a muscle cell, changes from a -70 to this positive -40 or somewhere around there. And that occurs then at the synaptic cleft at the neuromuscular junction. Okay, so the first step here, this action potential, this change in charge occurs at the end of the neuron, and it reaches the axon terminal. Okay, so here it is at the axon terminal. That causes voltage-regulated calcium channels to open. They aren't pictured here, but those voltage-regulated channels would then open, and then calcium would flood into the axon terminal. That is going to cause synaptic vessels, which have a neurotransmitter, acetylcholine, to be released into what's called the synaptic cleft here through the process of exocytosis. Okay, chemically gated channels are the lock and key sort of thing. You have the neurotransmitter binding to the receptor, which then opens the gate. Okay, it may open this gate, which then allows sodium to flood into the cell, which then causes an action potential. This occurs also at the synaptic cleft of the neuromuscular junction. So the next thing that happens, after the nerve impulse and acetylcholine is released, the acetylcholine diffuses across the synaptic cleft and onto receptors. Onto this is then the sarcolemma, the part of the muscle. That binds. That binding then triggers sodium to go through the channel, and that will continue until you have an enzyme, acetylcholine esterase, which will break down acetylcholine and then close that gate again. Okay, so here we have examples of both chemical and voltage regulated channels opening and allowing substances in. So excitation then occurs through these voltage regulated and chemical regulated channels. Again, it all starts with the stimulus from the neuron, which reaches the axon terminal. The voltage regulated channels then let calcium into the cell, which causes the synaptic vesicles to rupture. Acetylcholine goes across the synaptic cleft, binds to these receptors, which allows sodium to go into this skeletal muscle. That sodium is the excitation, the now change in potential or electrical signal, which then goes down the sarcolemma into the sarcoplasmic reticulum, which then releases calcium into the rest of the muscle fiber. About 80% of the calcium comes from the terminal cistern, 20% just comes from the sarcoplasm. And that then is excitation. The intracellular calcium then causes the troponin and tropomyosin to shift and expose the myosin binding sites on the actin, and the myosin heads can then bind with that actin. Okay, so now you have all the elements in place for binding, and that binding then goes through a process called contraction, where essentially the ratcheting motion of the myosin heads binding and then releasing and binding again all over the myosin pulls the actin and myosin closer together. So that is the contraction part. So there are a few positions that are formed. The first one is the cross bridge formation, where because the troponin and tropomyosin is moved out of the way, that binding site is exposed and binding occurs. The power stroke then occurs. This is where the movement actually occurs, and that causes the ADP and phosphorus to move away from the the myosin head. But now it's stuck there and it needs to move, and in order to do that it needs more energy. So to get it back down into the cocked position, it has to detach and has to use ATP in order to do that. It then needs more energy to move back, and so the cocked position then is hydrolyzing that phosphorus and creating ADP and P from the ATP. So, and then it goes back to the first. So this goes back in continuous fashion until the intracellular calcium levels go back down and the troponin and tropomyosin sites are blocked. And when that occurs, relaxation occurs. When the stimulus from the neuron ceases, the voltage-regulated channels on the axon close because of acetylcholinesterase. Calcium is pumped back into the sarcoplasmic reticulum using active transport. The troponin and tropomyosin move over and block the binding sites on the actin molecules. Okay, and that's how it works. That's how your muscles contract. It's a big, long process, but it follows a story. So if you can go from one end to the next, you can understand this pretty easily. Calcium is a big player in multiple sections of this process. The calcium ions initiate those voltage-gated channels to open. Calcium is also released by the sarcoplasmic reticulum and causes troponin and tropomyosin to move. Calcium pumps are reclaimed, or calcium is reclaimed as it's pumped back into the sarcoplasmic reticulum. And so if calcium isn't resorbed, then this process continues and those muscles stay contracted. And that actually happens in a process called rigor mortis. So after something dies, generally within the next few hours, the muscles will all contract and form the stiffness. And what happens is those dying cells are unable to exclude the calcium. They no longer have the ATP around to exclude the calcium. And ATP synthesis is also not there to move it. Since it's an active process, no ATP is around, you can't put the calcium back, so the muscles all contract. And so they remain in the power stroke position. Eventually, it all breaks down and it dissipates and the body becomes less stiff. A more live problem may be if there is some problem at the neuromuscular junction, which occurs in a condition called myasthenia gravis. And this is where you don't have enough acetylcholine receptors. And that's because of an autoimmune function. Essentially, where the immune system is attacking these receptors. So the symptoms of myasthenia gravis are all neurological in their behavior, neuromuscular at least. And so drooping eyelids, difficulty swallowing and talking, general muscle weakness are all muscle responses to neural input. And so when you have those problems, it may be a sign of myasthenia gravis. All right, so now moving more macroscopically, let's talk about the motor unit, which consists of one motor neuron and all the muscle fibers that innervate it. And you can see a motor neuron can split up into many different axon terminals, and and all the muscle fibers it touches are the motor unit. A muscle fiber is only innervated by one axon terminal, though. Okay, so you can't have multiple axons per muscle, but you can have one axon going to multiple muscles. Does that make sense? Hopefully. All right. A muscle twitch then is the motor unit response to an action potential of its motor neuron. So the response to that electrical signal. And it can be measured using what's called a myogram. So here we have tension, so how much force, and the time on the x-axis. The latent period is where cross bridges are not yet formed, even though that the signal has been sent, because it takes a little bit of time for that whole process to occur. The period of contraction then is where cross bridges are formed and ratcheting and going until you have a peak contraction. And then a period of relaxation will occur as that process resets, calcium's pumped back, acetylcholinesterase is at work. Different muscles have different times depending on enzymes and metabolic properties of those myofibrils, different types of muscles within there. Muscle twitches can also have different responses as you have different stimuli. Okay, and these can be measured using what's called a myogram. If you change the frequency of stimulation, so how often that you send an axon potential, you can come up with different types of responses. The first one is called wave or temporal summation. So essentially what you do here is if you excite the motor unit before it fully is able to relax, it will increase in tension, even though the actual stimulus itself has not increased. This can eventually lead to unfused or incomplete tetanus. Essentially, there's a maximum tension it can be at. And if there are bumps in that, then that would be unfused or incomplete tetanus. Complete tetanus is where you have this flat line here. Right? So this is complete tetanus, where the muscle is at full contraction. It can't contract anymore. That's the full force that it can exist at. There's another phenomenon called TREP, and this is where even after you allow the motor unit to fully relax, hitting it with another stimulus very soon after that relaxation will increase the tension. Okay, there is some point at which it levels off, but for some reason you have an increase in tension even though the stimulus has not increased. And the thought is this is caused by not all the calcium has fully reabsorbed back into the sarcoplasmic reticulum, even though it shows full relaxation, or some sort of warming of the muscle where those pathways are now open and more readily available. All right, so some of these, this would be wave summation here, this would be TREP here, and this would be complete tetanus. All right, so what then, looking at the whole muscle, accounts for the changes in the muscle contraction? Well, we can look at differences in the strength of the stimulation in the actual signal itself. Recruitment is how many motor units are being used. And this can be done by increasing the stimulus to affect more motor units. But there is a point at which there are some stimuli which are not enough to elicit an actual response from the muscle. So these stimuli, 1, 2, and 3, are too small, or at least 1 and 2 are, in order to even show an effect on the muscle or to recruit any of the motor units there. But after that threshold, then you will start to see more and more motor units being recruited. The sub-threshold stimuli then is where anything below there which will no longer show, which will not show a contraction. Okay, so 1 and 2 are part of sub-threshold stimuli. We have 3, which is the actual threshold, and then we have maximal stimulus. So at 7, okay, even if we increase the stimulus, we're already maxed out. We don't have any more motor units to recruit. And so that's the maximal. It doesn't do any good to increase the stimulus beyond 7 because we can't contract any more muscle fibers. Okay, the smallest muscle fibers are generally stimulated first, so they would be on this end. Okay, the largest are and least excitable are last, so they would be on this end. Okay, well, what are some clinical applications of this? Well, we talked about tetanus as a physiological function, but it's also a disease. Okay, there's a bacteria called Clostridium tetani, which is found in the soil that releases a toxin which causes your muscles to contract. It usually starts in the jaw, so it's sometimes called lockjaw, and then progresses to other muscles. It is actually not caused by rusty nails, although when people step on a rusty nail, they're like, oh, well, you better go get a tetanus shot. It's actually caused by soil microbes. Well, a lot of rusty nails are found outside in the soil, so that may be where the connection came from. So what voluntary muscles would be fatal if infected? Well, the diaphragm, which controls your breathing, would cause death. There are probably some other ones you can think of too. Vaccinations have made this pretty much non-existent. Getting a tetanus shot is an important thing, and over time you generally have to get a booster shot for that as well. Okay, so muscles and their energy are a big part of exercise science. We're going to talk about the energetics of muscle contraction. Within your muscle itself, there is energy being stored. So the myosin heads have ATP on them already. ATP doesn't have to get there. Even at rest, there's already ATP. So there's some energy already stored there, and that gives you a few seconds of contraction. Creatine phosphate is a substance which we didn't show, but it is very close to the muscles, has ATP there and ready, and offers about 10 seconds of contraction in a process called direct phosphorylation. But after 10 seconds, you're out of this immediately stored ATP. Creatine, you may see, is sold as some sort of muscle enhancing powder. Not sure if any of it works. So we have the myosin heads, we have the creatine phosphate, and then beyond there we've got glycogen, which is stored within the muscles, which is glucose molecules, which are all stuck together. That gives you 30 to 40 seconds of contraction immediately after. If you use glycogen in blood, glycolysis and the anaerobic pathway doesn't give you as much oxygen, but there is some immediately there. But the long-term solution for getting more out of it, so the initial steps give you 2 APT. But the full steps in using the Krebs cycle gets you 32 ATP. So if you have a little bit of time in order to break that down, it gives you a lot more ATP. Okay, there is some point at which you can't make it fast enough, and then you may revert to more of this quick anaerobic process. But for the most part, if you can get enough oxygen and you can get enough glucose, you can break it down for the full 32 ATP from the Krebs cycle and the electron transport chain. So different sports and different activities are going to have different processes which are important to that. So if you're thinking about those that need quick energy, the creatine phosphate, the stored ATP for the myosin heads, those are going to be mostly your powerlifting, right? Whereas exercises that require, more longevity, running around like soccer, those are going to be more aerobic. Okay, so if you're a sprint, heavy lifting, short-term energy requirement, you're going to use those shorter-term energy that is available. If you need more energy over a longer period of time, and endurance and aerobic exercise, you're going to utilize glycolysis in the electron transport chain and the full respiration. Beyond that, you can also use fatty acids, stored fat, to mobilize glucose or to mobilize molecules which can be used in the Krebs cycle and electron transport chain. Over time, though, you're going to cause what's called muscle fatigue, which is the inability to contract muscles even though you want them to, and that appears to be because of ionic imbalances. So at some point, the phosphorus, sodium, calcium, potassium, which are important for regulating those resting potential, just can't get back to normal. You can't flex those muscles anymore. Where does this excess phosphorus, sodium, calcium, potassium come from? Well, that goes back to when we were talking about the electrochemical balance which occurs in and outside of the cell. Caffeine, though, appears to be able to restore calcium levels to be released by the Sarcoplasmic Reticulum at pre-fatigued levels. So that's how caffeine is thought to be able to add energy to a system, even if it should normally be fatigued. All right, during extreme exercise, however, the body will shunt blood away from the muscles, towards the muscles and away from peripheral systems such as the digestive system. And this can lead to contradictions where, so your digestive system is no longer getting the nutrients it needs, and so you're going to barf, or perhaps poopy pants, which occurs in marathons. Not a pretty picture. We've all probably experienced going out and working out when we're not in shape. That first day is no fun because you get muscle soreness afterwards, delayed onset muscle soreness. So it's usually not immediately after the exercise, but a few hours to the day after. And this appears to be occurring from micro tears in the Z discs, micro tears in the fibers themselves. And then that causes an inflammatory response, which then presses on nociceptors or pain receptors, which makes it painful. But it's not by lactic acid buildup. And we know that because lactic acid is removed from the muscles hours after exercise, so it wouldn't make sense that that pain would continue if the lactic acid is gone. Here are some examples. This is a normal set of skeletal muscles. Here is delayed onset muscle soreness where those Z discs have torn. And here's another example here. Now when they grow back, they generally grow back stronger, bigger, often recruit more muscles, so a bigger muscle, a more fit muscle will replace it. There are a number of elements of recovery. Excessive post-exercise oxygen consumption. This is essentially being out of breath. What happens is the oxygen, you've run out of oxygen reserves and you need to replace those, so you'll breathe heavy until you do. You need oxygen energy to remove that lactic acid, and you need to replenish your glycogen stores, ATP and creatine phosphate stores as well. So all of that goes into, all right, well, I'm going to spend energy in order to get these things back to where they were. Heat production is also, of course, something you experience when you exercise. Only about 40% of the energy is used in these conversions of chemicals into energy, and so the rest is lost as heat. And so we sweat in order to radiate heat away from our body. Finally, there are injuries that can occur, including strains, tears, and sprains. A strain is where the muscle is stretched, maybe torn, also called the pull muscle, and the inflammation of that muscle then causes the joint to be immobilized. A tear is where the tendon, this is a complete tear, a smaller tear. Tendons don't heal very well, very quickly, so those can be severe. Sprains are where the reinforcing ligament is strained or torn. And again, healing on these takes sometimes a long time. I'll go over this example in class, but essentially my brother Joe ran a marathon, didn't go to the bathroom first, and it affected him, where he essentially had an upset stomach for a very long time after his marathon. So he didn't look very happy after he did this run. All right, we're going to talk a little bit about smooth muscle, although you will cover smooth muscle as well in the digestive system. Its structure is slightly different because the cells are spindle-shaped and of various different sizes. They have an endomysium, which contains blood, vessels, and nerves, very similar, and they're generally arranged in 2 layers, kind of crisscrossing pattern. The longitudinal layers run parallel to the organ, and they can cause dilation or increase in the circular organ. The circular ones run around the circumference, and they cause constriction of the circular organ and make it smaller. Peristalsis then is the alternation between those 2, circular and longitudinal muscles, which then causes kind of like a circle to go down the tube and push things out, kind of like how you would push toothpaste down up from the bottom. They kind of look like a Chinese, these circular and longitudinal muscles kind of act like a Chinese finger trap would, where you push on them and they shorten, and you pull on them and they decrease the diameter, although it's slightly different in their mechanism. The smooth muscle has structures similar to the skeletal muscle but different in function. They have varicosities, which are these bulbous endings rather than a axon terminal of the nerve fiber of the neuron. They have caveolae instead of T-tubules, which are extension of the sarcolemma. The actin and myosin is in a diagonal pattern rather than a horizontal pattern. And they have a molecule called calmodulin instead of troponin for calcium binding. They have dense bodies instead of the Z discs, which act as the anchoring point for the actin and myosin. They have much less sarcoplasmic reticulum, and they have a enzyme called myosin kinase, which phosphorylates the myosin or gives the ATP its power. So what happens in smooth muscle for contraction is the varicosities release the neurotransmitter across a much more spread out neuromuscular junction. An action potential is then generated across the muscle fibers into a synchronized contraction, so the fibers all contract at the same time. A voltage regulated channel opens calcium, which flows into the cytosol from the extracellular fluid, and most of it comes there rather than the sarcoplasmic reticulum. The calcium then binds to the calmodulin, okay, which then activates myosin kinase, that's MLCK here, which then activates the ATP on the myosin heads, which allows then contraction to occur until calcium is pumped back in to the sarcoplasmic reticulum and extracellular fluid. So again, these are the same sort of process as skeletal muscle, just the players are differently. They have varicosities instead of an axon terminal, caveoli instead of T-tubules. Most of the calcium comes from the sarcoplasmic reticulum in skeletal muscle and the extracellular fluid in smooth muscle. Actin and Myosin is in diagonal rather than horizontal pattern. They have calmodulin instead of troponin, dense body instead of Z-disc, and myosin kinase to activate those myosin heads. All right, and that's it for our muscles.